Hello student, in this video we are going to study regarding the sources of a crude drugs. So before start with the sources of the crude drugs, we know that crude drugs are nothing but the drugs which are obtained from the nature, where very least processing is carried out on these drugs. Okay, so the crude drugs are nothing but the drugs which are obtained from the nature with very less processing on them. Where these crude drugs we are generally obtain, or we get these crude drugs to obtain the different kinds of secondary metabolites. So we know that the secondary metabolites are nothing but the active constituents which are present in these crude drugs, and these are responsible for the therapeutic actions. Okay, so accordingly these crude drugs are obtained from the nature, from the different sources. So we will start with the classification of the crude drugs. So the first class is nothing but the biological sources. So whatever the biological sources which are present in the nature, they are going to be act as a source of crude drugs. So the first source of this crude drug is nothing but the plants. So we know that in the nature we can see the different varieties or numerous plants which are distributed in the different regions. So these plants are going to be act as a major source of these crude drugs, where the second biological source is nothing but the microbes. So microbes, these are nothing but the microorganisms like bacteria, viruses, rickettsia. So these are going to be acts as a source of a crude drugs. While in the biological source, animals, it is again one of the important class in for the sources of the crude drugs, where we know that the enzymes, hormones and different organ extractives, these are used as a source of the crude drugs. Then the second source of a crude drug is nothing but the marine sources, where we know that the different algae, sponges and the fishes are also going to be act as a source of a crude drugs. Then the third source of a crude drug is mineral sources. So different minerals, these are also used as a sources of a crude drugs. While the fourth one, that is the plant tissue culture. So it is a technique which is used for a development of new plant material by using a single cell. So that is known as the explant. So in this plant tissue culture technique, the different cultures, these are prepared, and that is going to be act as a sources of a crude drugs. Now we will start with the first source, that is nothing but the biological source, and the important one in this biological source is nothing but the plant sources. So we know that the plants are used for the treatment purposes, where these are used from a longer duration for the treatment of different kinds of diseases and the disorder like conditions. Okay, so the different parts of these plants, they contain the different types of chemical constituents or the active constituents, and these are going to show the different actions. So as per our traditional systems of medicines like Ayurveda, Siddha and Unani, we are using these traditional systems nowadays also for the treatment purpose, and we know that in these systems the most of the crude drugs, these are obtained from the plants and other natural sources. So accordingly these plants, these are commonly used as a biological source for the crude drugs, where the different parts of the plants, they contains the different constituents, and that is showing the different pharmacological actions. Now we will see regarding the examples. So the different parts of the plants. So first part we can say that is the rhizome. So in case of rhizome, turmeric, we know that turmeric, it is nothing but the haldi, and in our regular practices we are using this for different purposes, but it is mainly having the use like it is used for the antiseptic purpose. The second example is roots. So the example is of the root, that is snake root, which is also known as the Raulfia root, or it is also known as the Sarpagandha. So this root is commonly used as an hypertensive because it is going to lower the tension, or we can say the blood pressure. Okay. Then third example is flower bud. So clove is the example, that is nothing but the lavang. We are using its flower buds, which is having the carminative property. Carminative, that means these are responsible for expulsion of the gases. Then next example, that is flowers. So hibiscus flowers. So in Marathi it is known as the jaswant. So we are using this for the anti-ulcer property. While the leaves, so there are various plants of their leaves are used for the different purpose. So we know that tea leaves, then tulsi leaves we are using in our daily practices. Here the example is given of a vinca leaves. So this vinca, it is specifically having the anti-cancer property. Then the next example is the fruits, where the fennel fruit, that is nothing but the bari soap or the body shape. So this is generally used as an digestant, and it is again used as an carminative, and specifically it is used for its aroma, that is it is having the specific aroma which is because of the volatile. Then the next example is a bark. So cinnamon bark, that is known as the dalchini. So this is possessing the antimicrobial property, and we are using it as an spices. So this is regarding the plant sources of the crude drugs. The next biological source of the crude drugs is nothing but the microbes. So microbes are nothing but the microorganisms which are responsible or which are used as a source of a crude drug. So we know that these microorganisms which are responsible for causing the infection. So how these microbes are going to be act as a source of a crude drug, as they causes the infection. So how they are going to be used for treating the infection? So we will see now. So the different microorganisms like viruses, bacteria, not only that, but the rickettsia as well as some kind of toxins which are produced by means of these microorganisms. These are also going to be act as a source of crude drugs, where these microorganisms, they act as a source for many immunizing biologicals. So the immunizing biologicals are nothing but these are used for immunization purpose, and these are prepared from the particular type of microorganisms. Okay, so we will see the examples like viral vaccines which are prepared from the viruses. So the examples are rabies vaccine, smallpox vaccine, influenza vaccine, measles vaccines. So these are the examples of viral vaccines which are prepared from the viruses itself. Then second example, that is the bacterial vaccines, where the typhoid, cholera, plague and BCG vaccines. And these vaccines, these are prepared from the bacteria. Then the next example, that is the toxoids. So toxoids are nothing but the particular products which are prepared from that microorganisms only. Okay, so we know about the toxins. So toxins, these are formed, or this can be prepared by the microbes during their growth and the multiplication. So they produces endotoxins as well as the exotoxins. Endotoxins means the toxins which remains inside the cell of that microbes, and exotoxins, that is the toxins which are produced by the microorganisms, but they get spread outside the microbial cell. Okay, but these toxoids, these are nothing but the microbial products which are used to produce active immunity against that particular type of disease or the infection. So these toxoids, how these toxoids are formed? So the exotoxins which are produced by these microorganisms, these are treated with the formaldehyde, where because of such kind of a treatment their toxic properties, that is the toxicity of that exotoxins, it will get reduced or get completely eliminated. But their antigenic property means the property which is responsible for formation of antibodies as per the human immune response. So that antigenic property get retained in that exotoxins. Okay, so this formaldehyde treatment is carried out with this exotoxin to reduce toxic property and to retain the antigenic property, and these products are called as the fluid toxics. Then these fluid toxics, these are precipitated or adsorbed on aluminium hydroxide or aluminium phosphate. So whatever the adsorbed oxides these are produced, these are known as the adsorbed oxides, and these are possessing the antigenic property. For example, tetanus toxoid and diphtheria toxoid. So this is regarding the microbial sources of the crude drugs. So now we will start with the third biological source, that is nothing but the animal source. So we know that there are different kinds of animals which are present in the nature. So you are thinking that how these animals are going to be acts as a source of crude drugs. Here the animals, their products or particular hormones and enzymes which are prepared in that animals, these are used as a source of crude drugs, where some animals these are having the same physiology as that of the human beings, and certain enzymes and hormones these are going to be secreted by that animal similar to that of the human beings. Okay, so accordingly that enzymes, hormones or the animal products, these are going to be obtained that particular animal, and that is used for as a source of a crude drugs. Now we will see how these are going to be act. So these hormones, enzymes, animal extractive and some organs like bile, blood, pancreas, so these are used for the treatment purposes, where the examples of hormones, that is hormones are mammalian products and that are secreted by endocrine or ductless glands of the animals and released directly into the blood. So these hormones, these are obtained from the animals and these are used in the human beings which are having insufficiency of that particular hormone. So for example, thyroid. So thyroid glands of the sheep and the pigs, they produces the similar thyroid hormones, and that is used in the thyroid insufficiency treatment in the human beings. Where second example is of insulin, which is secreted by the pancreas of a cattle or a pig, and that is used as an anti-diabetic in the human beings. Then oxytocin, which is secreted by the pituitary glands of the cattle and the pigs, and this is used during pregnancy as it is going to cause the contraction of the uterine muscles. So this is obtained from the cattle and the pigs pituitary glands. Then the next hormone, that is the vasopressin, which is again obtained from the pituitary glands of the cattle and the pigs for the intestinal paralysis, while the gonadotropin, which is obtained from the horse serum and the urine of the pregnant woman, and that is used for the treatment of the different sexual disorders. Okay, so this is regarding the hormones. Now the second product, that is nothing but the enzymes. So enzymes, we know that these are responsible for carrying out different kinds of metabolic processes in the human body. So different kinds of these biological processes are carried out by means of these enzymes. So they act as an biological catalysts, where these are proteinous in nature and these are having the molecular weight from 13,000 to 8,40,000, where the enzymes like pancreatin, which is obtained from the pig pancreas and it is used in the treatment of pancreatitis. Pancreatitis, that means it is nothing but the inflammation of the pancreas which occurs because of some kind of infection to the pancreas. Then trypsin, which is obtained from the ox pancreas and that is used in the treatment of wounds and the ulcers. Where fibrinolysin, which is obtained from the human plasma, and this is used for venous thrombosis or pulmonary embolism. So this is used, fibrinolysin means this is causing the lysis of the thrombus or the emboli which is formed in the veins as well as the pulmonary arteries. Okay, so it is used for the lysis of this particular uh, thrombus which is responsible for blocking that veins. Then next is the pepsin. So pepsin, it is obtained from the pig stomach and this is used in the condition like dyspepsia. This means total absence of the pepsin. Okay, so the human which are having this dyspepsia like condition can be treated by means of using the pepsin which is obtained from the pig stomach. And the another animal source is nothing but the animal extractives, where the liver and the stomach preparations as well as the bile, it is used as a source of crude drugs, where the liver and the stomach are derived from the healthy and the domesticated animals, and these are converted into the suitable preparations which is suited for the human bodies, which are used as a transplant therapy in the pernicious anemia like conditions, while the bile, which is a natural liver secretion and that promotes the digestion as well as which causes the absorption of the fats inside the body. So accordingly, this bile is again obtained from the animals as an animal extractive and it is used as a sources of a crude drug. So this is regarding the sources of the crude drugs which is obtained from the animals. Now the next sources of a crude drug is the marine sources. So we know that the ocean and the seas, these are going to be act as a source of a crude drugs, where the different forms of algae or mosses or the sponges, as not only that but some fishes, these are also going to be act as a source of a crude drug. So the oil which is obtained from the fishes, it is also possessing some medicinal values and that is used as a source of a crude drugs. So this marine pharmacognosy, it is nothing but it is concerned with the naturally occurring substances of a medicinal value from the marine source, where it includes sponges, red algae, agar, etc. The ocean cover more than 70% of the earth's surface and it contains about 3 lakh invertebrates and the algal species. So most of them are going to be act as a source of a crude drugs, where these crude drugs obtained from these marine sources, they possess different therapeutic values such as antimicrobial agent, antibiotics, antivirals, etc. So we will see some examples regarding this. So antimicrobial agents and the antibiotics, for example, the cephalosporin which is obtained from the marine fungus, where antiviral compounds which is obtained from the Caribbean sponge, antiparasitic compounds which is obtained from red algae, anti-cancer agents which is obtained from Caribbean sponge, as well as the cardiovascular agents which are obtained from the marine algae and the Caribbean sponge, where these whatever the crude drugs which are obtained from these marine sources, these are having lots of therapeutic activities. Some are listed here, where they also possess the activities like anticoagulant activity, anti-inflammatory, antispasmodic activity, etc. So these marine sources, these are again a major source of the crude drugs. So the third source of a crude drug is nothing but the mineral. So we know that these minerals, these are metallic or non-metallic sources of the crude drugs, where the mineral sources, it includes kaolin, talc, diatomite, bentonite, fuller's earth, shilajit, asbestos, etc. Where the certain examples regarding these minerals, that is magnesium sulfate which is used as an purgative, then ferrous sulfate which is used in the iron deficiency, while the zinc oxide, it is used in the treatment of wound and eczema, as the zinc oxide acts as an skin protectant, therefore it is used in the treatment of a wound and eczema. So minerals, these are also and one of the source of crude drugs. Next important source of a crude drug is nothing but the plant tissue culture, and recently this technique is commonly used for the production of the crude drugs or for production of the secondary metabolites, where in this the in vitro cultivation of the plant cell or tissue or organ is carried out aseptically under controlled environmental condition, where for the growth purpose either liquid or a semi-solid nutrient media these are provided, where this technique is followed for the regeneration of a new plant and also for the production of primary and the secondary metabolites, where in this tissue culture technique, consider if a leaf is used as an sample for the tissue culturing, this leaf is cut into the smaller pieces, where after that its surface sterilization is carried out and that explant is taken, where it is inoculated into the nutrient media, where it is allowed to grow for a specific time duration, that is nothing but incubation, and that is results into the formation of a callus, where callus is nothing but the mass of the cells, and this mass of the cells further develops roots and the shoot, that is nothing but the organogenesis will occur, and it further results into the regeneration of the plantlets. So these plantlets are used for as a source of a crude drugs, while in some cases this callus is agitated for breaking into a individual cells, and these cells are then transferred into the bioreactor which contain nutrient media, where it is called as the suspension culture, and in this suspension culture these cells will grow and multiply, and which finally results into the production of the secondary metabolites. These secondary metabolites are then isolated and used for the medicinal purposes, where this tissue culture technique, it is commonly carried out for the large scale production of the secondary metabolites by means of using the bioreactors. Now here are some examples of the secondary metabolites. These are given. Digitalis leaves are used for the production of digoxin. Atropa bellatona is used for the production of atropine. Rauwolfia root is used for the production of a razor pen, and papayan is used for the production of the papayan, where this is regarding the sources of the crude drugs. If you like this video, please share this video, and if you are new to my channel, so please subscribe my channel to get more videos regarding this pharmacognosy subject. Thank you.